So we'll talk about the skeletal muscle tissue and how the contraction of skeletal muscle occurs, the molecular mechanism of contraction of the skeletal muscle. Uh, First, we'll see different types of muscles. Then we'll talk about the important functions of the muscles, and then we'll start the skeletal muscle. First, we'll see the structure of the skeletal muscle. Then we'll talk about the myofibrils. Inside the skeletal muscle, you have thin fibers. Those are called myofibrils. We'll see the structure of a myofibril. Then we'll talk about the contractile unit of muscles. Inside the myofibril, you have the contractile units of the muscles. That means the muscle contraction starts from those units, or it starts at those units. So we'll see what are those units in myofibril. Then we'll talk about the contractile proteins. Inside those units, you have some proteins, and the proteins form two types of filaments, thick and thin filaments. So we'll see how the proteins form those filaments and how those filaments work to contract the unit. Then we'll talk about a couple other things, cross bridging, neuromuscular junction, and clinical conditions. So those are the things we'll discuss in today's lecture. First, you already know that there are three types of muscle tissues present in our body. Skeletal muscle tissue, which is voluntary. We move our body parts by using the skeletal muscle. So, you know, we can move as we like. So that means skeletal muscle is voluntarily controlled. And cardiac muscle is a highly specialized muscle tissue or muscle which is only present in the heart, and it is involuntary, will not listen to you. It pumps the blood 24 hours throughout the life. And smooth muscle tissue is also involuntary. It will not listen to your command. It will perform its tasks by itself. So only skeletal muscle tissue is voluntarily controlled. Here you see how they look like under the microscope. We have talked about it before. Just quickly review. See the left one, the skeletal muscle tissue under the microscope. The fibers are cylindrical, long cylindrical fibers, number one. Number two, striations are present. Number three, multiple peripherally located nuclei. So in each fiber you have multiple nuclei, and those nuclei are not located in the center but peripherally located. Middle one is the cardiac muscle tissue under the microscope, cylindrical fibers. In case of cardiac muscle fibers, there are branchings. So you will see branches arise. Striations present, and another thing which is only present in cardiac muscle, intercalated discs, those thick bands. Usually single nucleus. The rightmost one is the smooth muscle tissue under the microscope. Smooth muscle fibers are not cylindrical. They are spindle shaped. That means like your eye, both ends are pointed and middle part is wide. And very simple muscle fibers, only a single nucleus located centrally, no striation, no intercalated disc, no branching, only a centrally located single nucleus. So those are three types of muscles or muscle tissue. Functions or properties. Properties of the muscle tissue. All muscle tissues have some characteristics or properties. What are those? Excitability, contractility, extensibility, and elasticity. Excitability is the ability to get excited. If you stimulate the muscle, it will respond to the stimulation, and that response is called the excitability, the ability to respond to a stimulus. And how the muscle responds? By showing contraction. That is contractility, the ability to contract or getting shorter. So the muscle shows the response by showing contraction, the ability to get shorter. Extensibility. If you pull the muscle from both ends, you can expand the muscle. So it is like, elastic thing that you use in your cloth. You can expand it by pressure, and that ability is called extensibility. And if you remove that pressure, it will go back to its actual shape. The ability to recoil, getting back to or going back to its normal shape, that is called the elasticity. The elastic thing that you use in your cloth, you can expand, right? That is the extensibility. Now if you leave that pressure, it will go back to its actual shape, right? That is the elasticity. So those are some properties or characteristics of the muscle tissue. Functions. With the help of the muscles, we move the body parts or bones. Circulation of body fluid. You know that cardiac muscle or heart contracts and relaxes, and that is the pumping action of the heart. And by the pumping action of the heart, blood circulates throughout the body. You all know that. So that is another important function of the muscle. Maintaining posture and body position. We use the skeletal muscles to maintain the body position. Stabilizing joints. Most of the muscles of your body cross the joints. And when the muscles, for example, you see here, this is your elbow joint, right? So biceps, triceps, all those muscles cross the elbow joint. Brachioradialis, biceps, triceps, and by crossing the joints from all different directions, the muscles stabilize the joints in your body. Heat production. You know that if your body gets too cold, shivering occurs, right? Frequent muscle contraction occurs. Why? Because muscle contraction produces heat. So that will increase your body temperature. So those are some important functions of the muscles in the body. Now we'll see the structure of the skeletal muscle. So we are now talking about the skeletal muscle structure. First, the whole muscle has a covering, and that is called the epimysium. So whole muscle is covered by a connective tissue membrane, that is called the epimysium. If you just see a muscle, for example, this is the biceps, okay? The muscle, whole muscle has a connective tissue covering that is called the epimysium. Okay, epimysium. Then inside the muscle you have column-like structures. So these columnar structures inside the muscle are called fascicles. So inside the muscle you have many fascicles, and each fascicle has again connective tissue covering, and that is called the perimysium. So each fascicle has the connective tissue covering that is called perimysium. Then inside the fascicle again you have smaller columns like this. These are called muscle fibers. Okay? Muscle fibers. Again, each muscle fiber has connective tissue covering. So each muscle fiber has covering, and the covering of the muscle fiber is called endomysium. Okay, so each muscle fiber is covered by endomysium. Again, inside the muscle fiber, this is so interesting, is second. Okay, so inside the muscle fiber you have thin filaments like this, many thin filaments, and those are called myofibrils. So you see how beautifully the skeletal muscle is structured. Columns within columns, in simple way you can say, right? Smaller columns within bigger columns, and this kind of structure gives the muscle its strength. You know, some big buildings nowadays are designed like this, like in Chicago, you know, the Sears Tower, columns within columns. Okay, first they make big columns, then smaller columns within that, then further smaller columns within that. That gives the flexibility and strength. So this is basically how the structure of a skeletal muscle is. So you see the smallest rod-like structures are myofibrils. So if I go from the smallest to largest, myofibrils, muscle fibers, then fascicles, and then whole muscle. Okay. Now if I just take one myofibril out from there, that means one of those dots, and see under the microscope, this is the myofibril. Okay, and inside the myofibril you'll see segments like this. It's very interesting, repeated in the myofibrils. So each segment is the functional unit of muscle contraction. So this is one segment, this is another segment, this is another segment, same thing, repeated, and each segment is called a sarcomere. So this is one sarcomere here to here, this is another sarcomere here to here, and sarcomeres are separated by Z discs. Okay, so the sarcomere is the segment between two Z discs. And when muscle contraction occurs, what happens? Since sarcomere is the unit of muscle contraction starts at the sarcomere. So when all sarcomeres contract at the same time, whole myofibril gets shorter. Now, inside the muscle fiber, when all myofibrils get shorter at the same time, the muscle fiber gets shorter. When all muscle fibers get shorter at the same time, the muscle fascicle gets shorter. When all these fascicles get shorter at the same time, the whole muscle contraction occurs. Make sense? So the contraction starts from the shortening of the sarcomere. Contraction of the sarcomere. Make sense? So that's how the muscle contraction occurs. Starts from the sarcomere. Now we will see how the sarcomere gets shorter. How the sarcomere gets shorter. Inside the sarcomere, you have two types of filaments, thick and thin filaments. Okay, so those are protein filaments. These are thin filaments, like this, and thick filament. These are thick filaments. Okay, so inside the sarcomere, you have thin and thick filaments. Okay, so this is thick filament. This is thin filament. We'll talk about that. Okay. Here you see the whole muscle, then muscle fascicle, then muscle fiber, and inside the muscle fiber you have the myofibrils. The same thing that I have shown here. Okay. Skeletal muscle fibers, as I have mentioned already, long cylindrical muscle fibers. The length could be up to 30 centimeter. So very long. Multiple peripherally located nuclei. Many mitochondria are present. You know, mitochondria are called the power houses because you need a lot of power to contract the muscle. That's why you have many mitochondria. And here you see just a muscle fiber and myofibrils inside. And now if you look carefully, inside the myofibril you have the sarcomeres segments. Let's see in here. You see just a small part of myofibril, and you see the Z discs, blue lines, and between two Z discs the area is the sarcomere. Okay, so sarcomere, and inside the sarcomere you see you have thin and thick filaments. Here by red lines they have shown the thick filaments, and by blue lines they have shown the thin filaments. We have thick and thin filaments inside the sarcomere, and those filaments help in the contraction of the sarcomere. Now the picture in the bottom, you see the thick and thin filaments. So now we'll see how the thick and thin filaments are formed. Okay. So the thick filaments are formed by the protein units. Those are called myosins. So each myosin unit or molecule looks like this, like a golf club like structure. Protein structure is myosin, and many myosin units bundled together, so like a bundle of golf clubs together to form the thick filament. So this is the thin filament. And thin filament is formed by two chains of actins. These balls are called actins. These are protein balls, and the actin balls form two chains, and that is the thin filament. Okay, so thick filament again is formed by the bundle of myosin units. These are protein units, and myosin looks like golf club, and thin filament is formed by the actins. Actin looks like a ball, and many actin molecules or units join together to form double chain structure, two chains, and that is the thin filament. Okay. Now, if you see a myosin, it is like this. This is the myosin. It has a head part, and a tail part. So, myosin has a head and a tail. Now, on the head of myosin, you have binding sites. One binding site is here, that is the site for actin. Okay. On the myosin head, you have a binding site for actin. Also, you have a binding site for ATP. Site for ATP on the myosin head. So those are two important binding sites present on the myosin head. On the actin ball, so this is the actin ball, you have a binding site. This is the site for myosin. So you see, on myosin head you have the binding site for actin. On actin you have the binding site for myosin. That means what? The myosin head will get attached to this, right? Okay. Now, during muscle contraction, the myosin head gets attached to the actin ball. But there is a problem. In relaxed condition, the binding site on actin is covered by a rope-like protein structure. That rope-like structure that covers the binding site on actin, that is called tropomyosin. So this is the tropomyosin. Okay, the rope-like structure that covers the actin, binding sites on actin. And on the tropomyosin, you have tiny protein structures just attached on the tropomyosin. These are called troponins. Okay. All these are protein structures. Actin, myosin, tropomyosin, troponin, all these are protein structures. Okay. Now, the problem here is, since the tropomyosin, that rope, is covering the binding site on actin, the myosin head cannot get attached to it. So first thing, during muscle contraction, what you have to do is move the rope away, so the binding site on actin ball will get free. Make sense? On the actin ball, the binding site will get free. So how that happens? To move the tropomyosin away or to the side, you need the calcium. That's why we say calcium is very important for muscle contraction. Calcium is needed to move the rope away. So what happens when you decide that you will contract your muscle, brain sends signal, and that signal releases calcium. And calcium enters into the sarcomere. Calcium ion gets into the sarcomere, and the calcium binds with the troponins. All calcium molecules will get attached to troponin. Okay? And that will move the troponins to the side. Like this, all troponins will move to the side, and that will pull the rope to the side, because on the rope tropomyosin you have the troponins attached. So when all the troponins will move to the side, it will move, that will move the rope to the side. Make sense? So the rope will move away from that place, and now the binding site on actin will get free. So what will happen? This is the actin ball, and this is the myosin head. They will get attached, and that attachment is called cross bridging. Okay? That attachment is called the cross bridging. That will happen. Very important. So you see, this is the actin ball, this is the myosin head, this is the attachment. Make sense? Then after that, what will happen? ATP will come and will get attached to the myosin head. So ATP will get attached to the ATP binding site, because you remember there is a binding site for ATP. So ATP will get attached to the myosin head. And now you see, this is the attachment. Now ATP will come and will give the power to move the head, and that's the flexion of the head or bending of the head. Make sense? That will occur, and that is called power stroke. Power stroke is the bending of the or flexion of the myosin head, and that is very important for muscle contraction. Now you see what happens. You have many actin balls. Power stroke is the bending of the myosin head. Who was asking? Okay, that is the power stroke. Okay, so now this is just one myosin, one actin, right? But in fact, you have millions of myosin heads and actin balls. Make sense? Inside the muscle. These are all molecular structures. So you have millions. So all myosin heads will bend, will get attached to the actin balls and will bend at the same time, like this. So now you see, this is the thick filament, has myosin and actin balls here. So all myosin heads will bend at the same time from this side, and myosin heads from this side will bend like this. So like this. So that will move the thin filaments towards each other from both sides, like this. Make sense? No, doesn't. Okay, good. So okay, so this is the actin ball. Make sense? Actin balls from thin filaments. You know that, right? Thin filament. So many actin balls and myosin head. Okay. So myosin head gets attached. Okay, and then moves like this. So what will happen? The balls will move this way, right? Balls are the parts of thin filament because they form the thin filament, right? So when all the balls will move like this, that means the thin filament will move like this. Make sense? From this side, same thing happens. So now you see, from both sides, the thin filaments will move towards each other. Does it make sense? Okay. And thin filaments are attached to the Z disc, both sides. So when you will move the thin filaments towards each other, the Z discs will move towards each other. Does it make sense? So what will happen? The Z discs will come closer to each other, right? And shortening of the sarcomere will occur. Make sense? So that's how the sarcomeres get shorter. So when all sarcomeres, just remember everything is working in fraction of second everywhere. So all sarcomeres at the same time will get shorter. Make sense? So whole myofibril will get shorter. When all myofibrils at the same time will get shorter, the muscle fiber will get shorter. When all muscle fibers will get shorter at the same time, muscle fascicle will get shorter. When all fascicles will get shorter, the whole muscle will get shorter. But everything is happening in a fraction of second. All bindings, power stroke, contraction of sarcomere, everything is happening in fraction of time. Yes. Do you have any question?
Oh no, I was just that's just why the muscle is basically softer before it contracts, and once it contracts, it all comes together.
Because in relaxed condition, tropomyosin, that rope is keeping the binding sites away from each other, right? So they cannot get attached. That's why contraction cannot occur. So when you release the calcium, you send signal to the muscle, calcium is released. That will start the process, right? So calcium will get attached to troponins. Troponins will move to the side. The binding site will get free. The myosin head is waiting for that. Will get immediately will get attached and will do what? Power stroke, right? So the thin filaments will move towards each other. The sarcomere will get shorter. All sarcomeres will get shorter at the same time. Yes. All these are proteins. Okay. So troponin, tropomyosin, actin, myosin. All those are protein structures. Okay. Now, these two, troponin and tropomyosin, these two are called regulatory proteins. And actin and myosin, these two are called contractile proteins. Okay. So actin, myosin, all these are proteins, but actin and myosin, those two are called the contractile proteins, and troponin, tropomyosin, these are regulatory proteins. Okay. So that's the mechanism of muscle contraction. Okay. Okay. So let's quickly review. Okay. So you have understood. Now you have to memorize. That is the important thing. Okay. So now tell me, the unit of muscle contraction is what? Sarcomere. Sarcomere is the part between two Z discs, right? And inside the myofibril, you have many sarcomeres, right? Segments. And inside the sarcomere, you have how many filaments? Two. Thick and thin. Thick filaments are formed by myosin or actin? Myosin. Myosin looks like what? Golf club. So thick filament is the bundle of myosin, right? Bundle of golf clubs. And each myosin unit has, or golf club has how many parts? Two. Head and tail. On the head you have what? Binding sites, right? One binding site is the site for actin and another for ATP, right? Those are two important binding sites on the head. Okay. Now thin filaments are formed by actin balls, right? They get attached to each other and form how many chains? Two. And that's why thin filament is a double strand or double chained structure. Now on the actin you have binding site for what? Myosin. On myosin you have binding site for actin, on actin you have binding site for myosin. So now you understand they will get attached to each other, right? Okay, but the problem is what? In relaxed condition, the binding site on actin is not free. Who covers? Tropomyosin. It is a rope-like structure, right? Rope-like structure. And on the tropomyosin, you have tiny protein structures, troponins. Okay. Now the task is removing or moving the tropomyosin away, right? To free the binding sites on actin. So who does it? Calcium comes from outside, right? And gets in the sarcomere and get attached to the troponins. Make sense? So when the calcium ion will bind with troponin, troponins will move to the side. Make sense? And that will move the tropomyosin to the side. Make sense? So the actin binding sites will get what? Free. Is it clear? And myosin head was waiting for that, right? So myosin head will immediately get attached. And that is called what? Cross bridging, right? And then after that, who will get attached to the myosin head? ATP, right? ATP is continuously being produced in the body, you know that. So ATP gives what? Power to do what? Move the head, right? That is called the flexion of the head. You know, flexion. Or bending of the head. And that is called what? Power stroke, right? Power stroke. So now you see all myosin heads at the same time get attached to all actin balls, right? And will do what? Power stroke from both sides like this. So that will move the thin filaments close to each other, and since the thin filaments are attached to the Z discs will come close to each other. They will pull the Z discs towards each other, and that is the shortening of the sarcomere. Okay? And when all sarcomeres will get shorter, the myofibril will get shorter, right? And so on. So that's the way how muscle contraction occurs. Okay. Okay, now the question is, from where the calcium gets in the sarcomere? We know that calcium is essential for muscle contraction. From where? On the myofibril, you have network of tubes. Many tubes are connected to each other and form network of tube-like structures. And that network of tube-like structure is called sarcoplasmic reticulum, or SR. And inside the SR, you have the calcium stored. So just around the myofibril, you have that network of tubes that is called sarcoplasmic reticulum, and it is filled with calcium. Filled with calcium. So that is the source of calcium. When you decide that you will contract the muscle, what happens? From the SR, calcium gets in. Now, who takes the calcium in? Another tube that brings the calcium inside the sarcomere from the SR, and that is called T tubule. That is a T tubule. So from SR, T tubule brings the calcium molecules inside and releases the calcium inside the sarcomere. Calcium will get attached to the trophon. Okay, so that is the source. So SR stores the calcium, T tubules bring the calcium inside when you decide to contract the muscle. Here you see the blue structure around the myofibril. That is the sarcoplasmic reticulum, network of tubules. Okay. Now, the mechanism of muscle contraction is called sliding filament model of contraction. Why? You already know. You see here, when the Z discs come close to each other, what happens? This is myosin, this is actin. So they move over each other like this. Okay, so if you see under the microscope during contraction, you'll see the filaments are moving like this. From both sides, the thin filaments are moving like this over the thick filament. That's why it's called sliding model of contraction. The filaments slide over each other. Here you see a sarcomere in two conditions, relaxed and contractile condition of the muscle. So we already know now that in relaxed condition, the Z discs are far away from each other, like the sarcomere is wider or longer. In contractile condition, the sarcomeres are shorter. So you can see here the dark lines in both sides, those are the Z discs, and in relaxed condition they are away. In contractile condition they are closer. Okay. Now one thing is, see here that this is the thick filament, and both ends of thick filament are connected to the Z disc by spring, protein spring-like structures. This is important, I may ask you. These are called titin, Titin. So the thick filaments are connected to the Z disc by protein springs. Those protein springs are called titin. Okay. So that is one thing you need to know. Attached to the both ends of thick filament, protein springs, thick filaments. Yeah, thick. Okay. So spring-like structure, protein structures connect the thick filament to the Z disc. Okay. Those are called titin. Now here another thing you see, that the space between two thin filaments is wider here in relaxed condition. When the muscle contracts, since the thin filaments come close to each other, the gap in between these two gets narrow, almost gone, because they move towards each other. And that's why under the microscope you will see the color of the bands changes. Why? Very simple. You see here, if you see this part under the microscope, just only look at this part. You have both thin and thick filaments, right? Together here. So more structures are here. And that's why under the microscope it looks dark. Here you have only thin filaments. That's why it is lighted. Light can easily pass. Here light cannot pass easily because you have more structures, right? That's why it is dark. Here you see kind of in between, because you have thick filaments only, no thin filament. Now, when the sarcomere contracts, this whole area has both thick and thin filaments. Okay, so that part is gone.
So that middle part is called an H then?
Yeah, different names, the bands, different. Since the structure, here you have only thin filament. Okay, here that is one area or one band. You don't need to remember these names. It doesn't make sense. Here you have both, so it is dark. Here you have only thick. That is another band. That is the H band, and again same thing. Okay? You don't need to remember. Just understand why you have those bands under the microscope because of the presence or absence of the thick and thin filaments. Okay, now we will talk about another important structure that is called a neuromuscular junction. The name is telling you it's the area where the nerve ends on the muscle fiber. That's why it is called neuromuscular junction. Nerve ends on the muscle or meets with the muscle. So you know that the nerves are formed by the neurons. So this is one neuron. This is the axon and the axon terminal. Axon terminal. And there are several axon terminals, and the axon terminals end on the muscle fibers. So this is muscle fiber, this is another muscle fiber, this is another muscle fiber. So axon terminals meet the muscle fibers or end on the muscle fibers, and that is the neuromuscular junction. These are the neuromuscular junctions where the axon terminals end on the muscle fibers. Now, if you see this structure, neuromuscular junction, under very powerful microscope, how it looks like, that we'll see. Okay, so we are just looking at this part, this tiny area. Okay, this tiny area. So, this is the axon terminal, this part, and this is the muscle fiber. Okay. So this is the neuromuscular junction. In the axon terminal, you have round membranous sacs. These are called vesicles. At the axon terminal, you have the vesicles. Inside the vesicles, you have the neurotransmitter. This neurotransmitter is called acetylcholine, ACH. Probably you have heard this name. Acetylcholine is the neurotransmitter inside the vesicles. And the part of the axon terminal that is facing towards the muscle, this area, this one, is called presynaptic membrane. Pre means before the synapse. So presynaptic membrane. And the membrane of the muscle fiber that is facing towards the axon terminal, this is called postsynaptic membrane. So presynaptic membrane in axon terminal and postsynaptic membrane on the muscle fiber. Post means after, pre means before. Postsynaptic membrane on muscle fiber. Presynaptic membrane in the axon terminal. Okay. Now, on the postsynaptic membrane, you have the receptors. these are called the receptors on the postsynaptic membrane. What kind of receptors? ACH receptors or ACH, acetylcholine receptors on the postsynaptic membrane. They will bind with ACH. That's why those are the receptors for ACH. Okay. Now this gap here, you see that axon terminal is not directly attached to the muscle fiber. That's why you see I left a little bit gap. So axon terminal is not physically attached to the muscle fiber. There is a gap, and that gap is called the synaptic cleft. You know, cleft, we use the term cleft, cleft lip, cleft palate, that means gap, right? So that is the synaptic cleft. There, the gap. Okay, so that is the structure. Now, when the electrical signal, you know that nerves carry electrical signal, we have talked about that. So very fast, the electrical signal, we send signal to the muscle, to the nerve. So electrical signal passes through the axon terminal like this, very quickly. And when the electrical signal passes through the presynaptic terminal, what happens? In presynaptic membrane, calcium channels get opened and calcium get in. Calcium channels get opened and calcium enters into presynaptic terminal. This calcium is different calcium than the calcium that comes from SR binds with troponin. That is different. This is another calcium. Okay, so calcium gets in, and the calcium causes rupture of the vesicles, burst of the vesicles. That's why you need calcium here, to rupture the vesicles. So when the vesicles will get ruptured by the calcium, ACH molecules will be released here. So these ACH molecules are now in the cleft, and these receptors are waiting to bind with the ACH molecules. So what will happen? The ACH molecules will bind with the receptors here, because these are ACH receptors. You can say ACH receptor complex or receptor molecular complex, we use that one too. So they get together. ACH binds with the receptor. Why that binding is important? This is like, you are putting your key in the lock of a door. Okay, so every day I put the key in the lock of this door, right? To do what? Open the door, right? So the ACH molecules are the key and the receptors are the locks. Make sense? So now the key is inside the lock, right? To do what? Open the doors. That is more simple. So you put the key in the lock to open the what? Doors. So many keys, right, are getting in many locks to open many doors or channels on postsynaptic membrane. So all those doors will get what? Open at the same time. Make sense? Open. Now, why the doors should be opened? When the doors on postsynaptic membrane get opened, sodium gets in. That is very important. Sodium ion gets in. Sodium gets in. That is very important. Why? When sodium gets in, sodium is positive. Inside the muscle fiber, the voltage will move towards positive. So now if I draw a graph, this is before the sodium got in, that is called resting membrane potential, okay? RMP, in resting condition. Now, after sodium gets in, inside the muscle fiber, it will get more positive because sodium is positively charged ion. So, this way if I draw positive, this way negative. When sodium gets in, I am measuring inside, so inside will move towards positive. That is called depolarization. Probably you have heard this before. Very important process. Depolarization occurs due to the influx of sodium. Make sense? Or entrance of sodium inside the muscle fiber. Sodium is positive or negative? Positive. So when positive gets in, inside the fiber will become more positive. That's very simple, right? So we'll move up because I'm drawing positive that way, right? It will move up. That is called what? Depolarization. Very important process in physiology.
So what is sarcomembrane? Does it own
Which one? Sarcolemma is the membrane that's covered the sarcomeres. We are not talking about that. So depolarization. Now the question is why you need depolarization? Why that should happen? Right? That is the question. Why we need to know depolarization. You remember, let's go back. When muscle, we decide that we will contract the muscle from SR. Who gets in the sarcomere? SR stores what? Calcium, right? Calcium, right? So calcium gets in, right? And calcium binds with troponin, right? Troponins will move to the side, that will move the tropomyosin to the side, will free the actin balls, right? All these things will happen. But we don't know yet who will give signal to SR to release calcium. Depolarization. Okay? So when the muscle will get depolarized, then the sarcoplasmic reticulum or SR will release the calcium into the sarcomere. So that's the signal to release what? The calcium. Make sense? So now you got everything. Now see, when we decide that we will contract the muscle, we send signal from the brain to the muscle, right? That signal is the electrical signal. Make sense? Electrical signal that will travel very fast through presynaptic terminal, and that will open the calcium channels. Calcium will burst the vesicles to release the ACH. ACH will bind with the receptors on postsynaptic membrane. Is it clear? So the doors will get opened because you are putting the key in the lock, right? And who will get in? Sodium. Sodium is positive, right? So inside the muscle fiber will move towards positive. That is called what? Depolarization. That when depolarization will occur in the muscle, that will give signal to SR to release the calcium. Calcium will now bind with troponin, will move the troponins to the side, will move tropomyosin to the side, actin balls will get free, myosin head will get attached, and will do power stroke. Make sense? So that is, now we got the complete picture. So if I ask you in the test, this is very important structure, draw and show the parts of a neuromuscular junction. This is a neuromuscular junction, okay? So you practice this one, so that will be helpful. Yes?
Sodium was in the vesicles?
Sodium is everywhere in your body, not in the vesicles. Vesicles contain what? ACH, right? Sodium and calcium are in the space here, in cleft. Cleft has fluid. Everywhere in the body you have fluid, right? Everywhere. So in the cleft you have the fluid. In that fluid you have the calcium, sodium, all those ions. Okay, make sense? Okay. So just remember those things I have explained you. That should be good enough. Okay, now we'll go over and try to understand what's going on. Do you have any question?